Good afternoon, ladies and gentlemen. We cordially welcome you all to the 3-minute presentation sessions and the certificate awarding ceremony of the 3rd Undergraduate Research Symposium organized by the Faculty of Veterinary Medicine and Animal Science, University of Peradeniya. I would now like to invite the following dignitaries to light the oil lamp to start the afternoon proceedings. Vice Chancellor of the University of Peradeniya, Professor M.D. Lamawansa, the Dean of the Faculty of Veterinary Medicine and Animal Science, Professor P.G.A. Pushpakumara, one of the deans representing deans of other faculties, Professor Sarath Kodituaku, the keynote speaker of today's event, Dr. M.R.A. Priyantha, Dr. Sampath Lukugalapati, representing all the heads of the departments of the faculty, Chairperson, Veterinary Medical Education Unit, Professor Ruwani Kalupahana, Chairperson, Faculty Research Committee, representing all academic staff of the faculty, and one of the student presenters. I now invite Professor P.G.A. Pushpakumara, the Dean, Faculty of Veterinary Medicine and Animal Science, to deliver the welcome address.
Good evening to all of you. Before I extend my formal welcome, please allow me also 3 minutes to briefly outline the history of the undergraduate research project within the faculty and provide overview of the Undergraduate Research Symposium 2023 and explain the process we have followed to select today's presenters. The student research project was introduced as a partial requirement for completing the BVSc degree during the curriculum reform in 1993. However, the current research project does not carry any credit value and start usually after semester 5 or when the students are entered into the 3rd year. And some students start their project even maybe late in the semester 6 or 7. And we allocate period of 5 to 6 weeks for completing their project following the final BVC examination. In response to this limitation, our new curriculum introduced in 2020 allocates 6 credit to the student research project and mandates its completion before entering the lecture-free final year. The Undergraduate Research Symposium in its present format began 2 years ago and today marks the 3rd Undergraduate Research Symposium in this format. Prior to this transformation, it is solely featured presentation of student research projects. This year, student project presentations were categorized into 6 thematic areas, namely farm animal production and health, companion animal health, poultry production and health, pharmacology, food and animal feed, wildlife and aquaculture, and the first time in our research symposium, the veterinary education and extension. So veterinary education is becoming very important part of our research program in our curriculum. This presentation took place on September 4th, and we assessed by independent panels, each responsible for selecting the best presentation within their respective themes. The chosen 15 students will now present their project finding within 3 minutes time frame. Now let me extend a warm welcome to the dignitaries, this important faculty ones. Firstly, I would like to extend my warm welcome to Professor M.D. Lamawansa, the Vice Chancellor of University of Peradeniya, for gracing us with this presence today. Our VC has constantly shown unwavering support for research, be it at the undergraduate, postgraduate to academic levels. His initiatives have also resulted in the allocation of funds for multidisciplinary research within the university. I would also like to offer hearty welcome to our keynote speaker, Dr. Roshan Priyantha, Senior Scientist of Veterinary Research Institute, for graciously accepting our invitation and being here with us today. Furthermore, I extend a warm welcome to our panel of judges, which include Professor Fazia Noordin, Professor Kosala Marabe from Faculty of Medicine, and Professor Ruwan Jayasinghe from Faculty of Dental Sciences, and Senior Veterinarian Dr. Vipula Dharamardana. I would also like to welcome the deans of the faculties, namely Professor Kodituakku, Dean, Faculty of Agriculture, Dr. Manjula Artigula, Dean, Faculty of Dental Sciences, and the Dean of the Faculty of Management. I would also like to extend my warm welcome to heads of the departments, all academic and non-academic staff members of the faculty, and other invited dignitaries who have joined us for the Undergraduate Research Symposium 2023. Last but certainly not least, extend my heartiest welcome to the presenters and students of the Faculty of Veterinary Medicine and Animal Science. And I would like to take this opportunity to thank also the VMU, Dr. Lalanthi, who is heading he's the chairperson at present, and also all the members supported to organize this Undergraduate Research Symposium of 2023. So thank you very much.
Thank you, sir. I now take this opportunity to invite Professor M.D. Lamawansa, the Vice Chancellor of the University of Peradeniya, to address the gathering.
Good afternoon, Dean of the Faculty, Professor Pushpa Kumara, and Acting Deputy Vice Chancellor, Professor Sarath Kodituakku, the other deans, academics, and the judges are the most important people today. And dear students, ladies and gentlemen, and thank you very much, Professor Pushpa Kumar, for inviting me. And also, I'm very glad to experience this kind of activities. And if you look at recently some of the, I think every day there is something related to research, international conference or something of this nature. So this will certainly help to improve the university's research outlook. In 3 minutes, dear students, and if you can explain your research to a layman who do not know anything about it, the concept behind, the methodology, and the results, and also what you expect in the future, and then you have achieved your targets. When I was a dean back in 2013, and I told my academic staff, and we had few thousand rupees coming from somewhere, we will get you to make your research presentations, the proposals 5 minutes each. There was an uproar in the faculty, how can we do this in 5 minutes, and we need at least 15-20 minutes to make an impact. I said, if you cannot do it in 5 minutes, do it in 3 minutes. But we did it in 5 minutes. 5 minutes, that was the experience that I had. And dear students, all the best for your presentations today. Thank you very much.
Thank you, sir. And sir, can we please ask you to ceremonially launch the proceedings of the 3rd Undergraduate Research Symposium? Thank you, sir. Now, I would like to introduce our distinguished panel of judges, Professor Ruwan Jayasinghe, Professor Fazia Noordin, Professor Kosala Marabe, and Dr. Vijitha Dharamardana. Now it's time for you all to witness the presentations. May I now kindly invite Mr. W.A.S.D. Veerasurya, and the title of the presentation is, Yes, We Are Noisy Neighbors.
Have you ever thought the noise of your vehicle can affect the life of wildlife? So I studied about the effect of anthropogenic noise on predator awareness in yellow-billed parrot. Because of the increased urbanization over last decades, the background noise level in the environment has increased drastically. Since the life cycle of birds mainly depends on the acoustic communication, from their parent-offspring communication and their mating behavior, and most especially the antipredator behavior depends on the acoustic communication. This increased background noise level can affect them very badly. So how does this can happen? This can happen by distracting them from identifying these predator calls or the alarm calls of the predators because of the increased background noise. And this can happen also by directly identifying these noises or traffic noises, horns or the musical notes as predators. Next thing is noise masking. What is noise masking? Noise masking is hiding small natural sounds because of the increased background noise level. So my question was, is there any difference between their responses to predators or the time they have taken to respond to predators when there is anthropogenic noise? To study this, I conducted playback experiments. I used shikra as my predator species because shikra is a common predator for the yellow-billed babblers. Then I combined these shikra calls with the traffic noise to make the effect of the anthropogenic noise. Then I used spotted dove as my control species because spotted doves and the yellow-billed babblers were commonly seen in the environment, and I thought there is no specific information contained in the yellow-billed babblers for the spotted dove calls. So I conducted playback experiments and observed their behavior. What did I found? Yes, as you thought, the time they have taken to respond for the predator calls when there is anthropogenic noise, there is a significant difference. When there is anthropogenic noise, time they have taken to respond to predator calls has increased significantly. And when there is no anthropogenic noise, the time they have taken to respond is much lower. So it confirms, yes, we are noisy neighbors. What do you think? Is there any possible effect on their survival because of this anthropogenic noise? Thank you.
Thank you. And may I now kindly invite Ms. Kumudumali WU and the title of the presentation, A Superbug Resistant to Carbapenem Resistance Escherichia coli.
Let me ask you something. Do you believe the antibiotics have saved billions of lives? Yes, they have. Unfortunately, we are at the verge of that era where these antibiotics successfully treat the infections. The bacteria, they are wonderful evolutionary machines. Every time they are going to find a way out of antibiotics and to develop resistance to it. What do you think? The word antimicrobial resistance has introduced. Now it is a global health threat. It is predicted by 2050, drug-resistant infections will kill 10 million people a year. How we are going to stop it? Are we going to develop new antibiotic? Will it be useful? The thing is, the newly developed antibiotics also eventually will stop working. If you can remember, the urinary tract infections earlier treated successfully with the antibiotics, the first discovered antibiotics called penicillin. But now we are at the last resort of antibiotics, carbapenems. Take a look at that image. The bacterium has become superior and the carbapenems has become less useful. Why is that? The carbapenem resistant bacteria is there. My research focused to identify the development mechanisms of carbapenem resistance. I evaluated several of these superbacteria with the established model, with the established methods, and we found these superior bacteria has some kind of superior enzymes that may use to fight with these carbapenems. And we closely look on the genome. There are some genomes, there are some genes of this bacteria that has changed. So, surprisingly, we have found These superior bacteria has developed resistance to at least 15 antibiotics. It is probably a superbug. And take-home message is here, the scientist can evaluate the antimicrobial resistance, but we have a responsibility to find a way to combat this resistance. Thank you.
Thank you. May I now kindly invite Mr. G.T.V. Ananda, and the title of the presentation is Infectious Laryngotracheitis in Sri Lanka.
Good evening, everyone. Even though many of you have heard the term ILT, some of you may be wondering what is ILT. So let me give you a brief introduction about ILT. ILT, or infectious laryngotracheitis, is a highly contagious respiratory disease in poultry caused by Callid herpes virus 1. So what do you think? Can ILT be present in Sri Lanka? Even though ILT has shown a worldwide distribution, when it comes to Sri Lanka, there is a lack of comprehensive data about the current disease situation in our country, except for few studies. For instance, a study done in 2022 has proven the first etiological evidence of ILT in our country. Furthermore, our field veterinarians have stated that the frequently encountered respiratory cases in poultry are quite difficult to differentiate because of the non-specificity of clinical signs. And it is very important to note that there is no current vaccination protocol for ILT in our country. So after considering all those factors, I firmly believe that ILT virus is present in Sri Lankan poultry population, but it is being largely underdiagnosed due to lack of facilities. So with that in mind, the study intended to detect whether ILT virus is present in Sri Lankan poultry population and to find out a rapid diagnostic method to detect this virus in laboratory conditions. So to do that, a total of 18 tracheal and lung samples were collected from dead birds showing respiratory signs from Kurunegala and Kandy districts, and DNA was extracted from those samples and subjected to PCR. And as you can see here, interestingly, only 3 samples from Kurunegala have yielded positive results, which indicates the overall prevalence of 16.67%. But when you consider district-wise, Kurunegala district has given 33% prevalence. So let's focus about the elephant in the room here. So as my study finding shows, the ILT virus is present in Sri Lankan poultry population. So what could be the possible reason for that? The most likely reason for that is the absence of a vaccination protocol in our country. Even though some commercial level farmers import this vaccine and use this vaccine in their farms under user permit, that vaccine is not available in local market. And since this is a viral disease, the preventive measures like biosecurity measures are very important. But most of our local farmers face financial constraint to implement these biosecurity measures. So it is high time for the relevant authorities to implement a vaccination protocol and encourage the farmers to improve their biosecurity measures to combat future ILT outbreaks. So finally, just think, would you like to see a huge economic impact and huge economic loss in our poultry industry in the future? Thank you.
Thank you. And now I invite Ms. D.A.H.Y. Senarath, and the title of the presentation, The Brain Behind the Veterinary Academic Success.
You all have probably heard about IQ. You may have gone through several IQ tests or may have purchased some books to improve your IQ knowledge when you were a small one. So, IQ is the raw processing power of our brain. In the world of academia, we often use the word IQ as the major determinant of academic success. But here's the thing. Let me say that there is another major influential factor to the academic performance which is named as EI or emotional intelligence. Let me introduce these two words to you in a brief way. IQ. Well, IQ is just like having a small super fast computer inside your brain. It may help you to solve complex problems as well as to effectively face the exams. The emotional intelligence is the ability to manage, utilize and effectively navigate and perceive the emotions of one's own and the others in the social interactions. So, that is the emotional intelligence. So, let us move back into our project. What we have done is to see the intricate relationships between IQ and EI towards the academic performance of the veterinary undergraduates in Sri Lanka. We have used a sample of 100 veterinary students and we have given a standardized IQ and EI test to each of them and evaluated their results. We have used standardized statistical tools to analyze the data and the results were amazingly like this. There was a strong positive correlation between the academic performance and the EI and IQ, and also the academic performance is predicted by IQ and EI, and we have derived a regression equation predicting the academic performance from the IQ and EI. It was like this. What is the mean of these findings? Let me just tell this in a simple words. IQ and EI are just like the two wings of a bird. Without the IQ and EI or one or the other, the journey towards the success in academic, journey towards academic success will not be achieved. So, what does this mean to our curriculum? What we can improve, how we can improve our curriculum through this? By incorporating curriculum adaptations, we can improve the emotional intelligence and intelligence quotient of the veterinary students of our faculty. So, let us empower a balanced education which improves the empowers the brain and our hearts towards the academic performance of veterinary students. Thank you.
Thank you. And now I kindly invite Mr. Wesley J. And the title of the presentation is Kandy District Oriental Theileriosis.
Have you ever heard about Oriental Theileriosis? Theileriosis is a major concern in the livestock industry in Sri Lanka. It is caused by protozoa. Infection occurs in the cattle, goat, sheep and buffaloes. Disease transmission occurs through the infected ticks. There are two types of Theileria species in Sri Lanka, Theileria annulata and Theileria orientalis. Oriental Theileriosis is caused by Theileria orientalis and it is a subclinical disease and it has negative impact on the farmers such as reduced milk production and weight loss, abortion and death of the animals. The prevalence of Theileria orientalis among cattle in Sri Lanka was around 50%. The wet zone has higher prevalence than the dry zone. The previous studies shown that the prevalence in the Nuwara district was 98% and in Galle it is 100%. Both are from the wet zones. But however, no more studies have been conducted in Kandy district. So, we selected Kandy district for our sampling. There are 20 veterinary ranges in Kandy district. So, the cattle population in the Kandy district is 24,000. So, we plan to take 380 samples for the studies and also we plan to do the genetic sequencing. But due to the economic crisis, fuel crisis, and due to the limited time period, we only selected 3 veterinary ranges and 60 samples were selected for the sampling. And we have done the blood smear and PCR studies. The results indicated that only 2 samples were positive for the blood smear and 4 samples were positive for the PCR. PCR is more accurate than the blood smear identification due to its high sensitivity, high specificity, and early detection. Our results indicated that only 7% of the sample tested positive. This low positivity levels due to low prevalence of ticks in those veterinary ranges, or a good tick control management practice, or semi-intensive and intensive management system. There are 11 genotypes of Theileria orientalis. Genetic sequencing or genotype specific PCR is required to find out the genotypes of the species. Furthermore, studies are required, and we have to find out the prevalence and genotypes of Theileria orientalis among cattle in Kandy district. Thank you.
Thank you. Now I kindly invite Mr. Pune K.A. and the title of presentation, Use Smart, Do Smart and Be Smart.
Let me guess, how many of you have smartphones? I think most of you have smartphones. This is the smart world, so smartphone is most of with you. Let me guess, who is a smart pet? A smart pet is a person who is going to rapid diagnosis in a very short period of time and accurate diagnosis and go for their treatment promptly. Okay. Now let's see how a smart pet works. Let's take an example. In a smart pet, when case presented to him, he will we'll take a case. Okay. A dog with a cutaneous mass will present it to him. What will he do? He will do fine needle aspiration and observe under the microscope. When diagnosing that fine needle aspiration, he will face some difficulties. What are the difficulties? Sometimes there's a problem in diagnosing it as a cancer or other type of condition. Okay. Now what will he do? He has to ask help from other outside pathologists. Okay. When transporting those prepared slides and other stuffs to the pathologist and taking diagnosis, it will take more time, and this will delay the diagnosis and getting treatments. Okay. Now what he can do? He can use another method for quick transmission of those prepared slides. Okay. This is my student project was. I thought about new concept. Everyone has smartphones, and using smartphones, they can capture those images. For that, I took round cell tumors in canines and prepared cytological preparations from those lesions. After that, I captured images from those cytological preparations. After that, those cytological preparations were sent to overseas pathologists. They gave me results within few minutes to hours. This method I use telecytology. It is a very good method that we can transfer images. I transferred images through internet. After that, they gave me result with few times, and I analyzed them with histopathology as a gold standard test. It gave me really good result. When comparing with histopathology, it has very good diagnostic accuracy. Then I compared it with statistical. When comparing it with statistically, it has good statistical significance and low interpathologist variability. When diagnosing round cell tumors, I think telecytology is a good method. We can further improve our test to diagnosing other type of cancers and tumors. This will make our technology and combine with our cytology and make good results. So we can finally use our smartphones, capture cytology, and be smart pets. It will be a good concept in future. Thank you.
Thank you. And now I invite Ms. G.I.U.S. Satsarani, and the title of the presentation, Elephants and Us, Behavior of Orphaned Elephants in the Presence of Humans.
In a situation where we hear deaths of wild elephants in a regular basis, the need for rehabilitation of elephant cows that become orphaned in the wild becomes really important. Udawalawe Elephant Transit Home offers a rehabilitation program for elephant cows which become orphaned in the wild with the intention of releasing them back into the wild after the rehabilitation. These elephant cows are managed as a herd of wild elephants with minimal human exposure, and they are exposed to humans during the milk feeding sessions which are held within the public view. I studied whether the human exposure can cause stereotypic behaviors among elephant cows during their milk feeding sessions which are conducted in the public view. So, what are stereotypic behaviors? Any repetitive movements that are performed by elephants without a specific function or goal, like trunk tossing, head bobbing and swaying, could be considered as stereotypic behaviors. These behaviors are mostly related to stress, and these are mostly discussed in elephants under captivity. So, in my study, I selected two sampling sites as the waiting area and the feeding area. So, the feeding area is the feeding station where these elephants are fed with milk in front of the public, and the waiting area is where they wait before they are sent for milk feeding at the fence gates. According to what I have observed, the elephants were showing repetitive behaviors only at the waiting area, and trunk tossing was this only repetitive behavior, and it was mostly seen related to the waiting area. So, this trunk tossing behavior was also not classified as a stereotypic behavior, as it was appeared to be exhibited in anticipation of drinking milk. And moreover, I looked at the age and sex specific behaviors of these elephants during their milk feeding sessions. So, I saw there was a significant effect of their age and the interaction of their age and sex on this running behavior at the feeding station, and there was a significant effect of their age and their sex on the walking behavior at the waiting area. And thus we can come to a final conclusion that these elephant cows do not exhibit any stereotypic behaviors during the milk feeding sessions which are conducted in the public view. Thank you all.
Thank you. And now I would like to invite Mr. Thanis S. And the title of presentation is Unveiling Compassion, Veterinary Students and Euthanasia.
Unveiling Compassion, Veterinary Students and Euthanasia. Here you can see a suffering dog with irreversible pain and incurable disease. So what will you do if you handle a case like this in your veterinary profession? The same question was asked to the students of the Faculty of Veterinary Medicine and Animal Science. So the answer were 63% of the students who said yes, we will consider euthanasia as an option when there is no other alternatives to cure the animal. Here you can see, very interestingly, gender emerged a significant factor that influencing euthanasia attitudes. Here you can see 56% of the females said yes, almost half among them. But you can see males who are 82% of them said yes to euthanasia. So which means males are very higher in considering euthanasia as an option than females. The odd ratios were 3 times higher. These are the students who are going to be professionals in a short time period.
So in future, in making decision on animal euthanasia, male veterinary surgeons are the mostly ones who are going to take the decision on that. The females are very hesitated to make a decision on animal euthanasia. And the other thing is, during the semesters, students in advanced semesters who said yes to euthanasia, but students in early semesters, first year, second year, they said no to euthanasia. This is due to the knowledge and clinical exposure is increasing through the academic years. And the other thing is, in other countries like European Union, USA, they have well described laws and legislations about animal euthanasia. They have good technical standards. But Sri Lanka is in infancy level. We have to have good technical standards of animal euthanasia, and we have to have good laws and legislations. And finally, awareness and knowledge and education of the animal euthanasia have to be given not only to the veterinary undergraduates and the veterinary professionals, but also the farm and pet animal owners. Thank you.
Thank you. And now I would like to invite Ms. J.W.Y. Seniviratna and the title of the presentation, Subclinical Prevalence of Babesia bigemina in 3 veterinary ranges.
Good evening, all of you. Do you know about bovine babesiosis? Bovine babesiosis caused by Babesia bovis and the Babesia bigemina, mainly in Sri Lanka. European cattle breeds are mainly vulnerable to the infection. In Sri Lanka, ruminants are highly susceptible to the infection, such as cattle, zebu, and buffaloes. Main clinical symptoms of the bovine babesiosis are high fever, anorexia, depression, constipation, dehydration, hemoglobinemia, and hemoglobinuria. In my study, mainly I focused on the subclinical molecular prevalence. So I have selected 3 veterinary ranges. In bottom photograph of my presentation show these 3 veterinary ranges. I have collected 60 blood samples and blood smears among the cattle in these 3 veterinary ranges, and I have done molecular assay and the blood smear analysis. According to the blood smear analysis, all the samples were negative, but PCR assay 1 out of 6 samples were positive. According to the old result, subclinical molecular prevalence of the bovine babesiosis among these 3 veterinary ranges are very low. There are a lot of reasons for low prevalence. They are we collecting the samples during the dry season, and most of the farmers practice intensive managed system with cut and fed feeding system, and low tick prevalence in those regions. According to the previous study also, Babesia bigemina prevalence is very low in Sri Lanka. So due to the due to lack of transportation facilities, due to lack of funding, sample number was limited. So to get a better understanding about the subclinical molecular prevalence of the bovine babesiosis, sample number should be increased. Thank you.
Thank you. And now I would like to invite Mr. U.D. Jayasekara and the title of the presentation, Molecular Detection and Phylogenetic Analysis of Fowl Adenovirus Serotypes, present in northwestern, western, and central provinces of Sri Lanka.
Good evening, everybody. My topic is molecular detection, serotyping, sequencing, and phylogenetic analysis of fowl adenovirus in Sri Lanka. So, fowl adenovirus is highly emerging disease, and also it is an infectious disease causing high burden to poultry industry, especially in broilers in Sri Lanka as well as in the world. So, when we consider about the FADV detection in molecular level, it is very important in terms of controlling and preventing the disease. So I selected 52 postmortem samples from suspected cases in field from the fields and prepared 11 pooled samples using that. And then I extracted DNA using commercially available DNA extraction kits and analyzed the quality of the DNA. It means purity and concentration of the DNA using nanodrop spectrophotometer. And then those samples were subjected to PCR using FADV specific hexone gene primer, and then followed by agarose gel electrophoresis, and then visualized under UV light. And you can see the results of that in figure number 1. All the samples I have tested was negative for FADV originated DNA. So therefore I continued the study using previous study positive samples, and those samples were submitted to DNA sequencing by Sanger sequencing, and then after that analyze the Sanger sequencing results you can see in figure number 2, and then those forward and reverse sequences were pooled and aligned together, and final sequences were obtained as shown in figure 3. And using those final sequences, we blast in at NCBI gene bank to get most related sequences from NCBI gene bank as shown in figure 4. And then after using those final sequences in figure 4, we aligned local sequences and also from sequences from NCBI gene bank together and prepared aligned sequences as shown in figure 6. And then using those sequences, I selected the best model to perform a phylogenetic analysis using Mega 11 software as seen in figure 6, and using local sequences and sequences from NCBI gene bank, I prepared the phylogenetic tree. You can see it in figure 7, and red arrows indicate where the local isolates represent in the gene tree. And after that, two sequences from Sri Lankan isolates were deposited in NCBI gene bank, which is shown in figure 8. And also samples were positive samples were further analyzed using serotype specific primers, and we found that serotype 2 and 8B was from those samples, and this is the first time in Sri Lanka we have performed the sequencing and serotyping for FADV virus. Thank you.
Thank you. And now I would like to invite Ms. S. Thiruthanigasalam, and the title of the presentation is Reap What You Sow.
Good evening, all of you. Today, within these 3 minutes, we are going to discuss about a thing that we got due to our own activities, and that is antimicrobial resistant. So, for this study, water samples from selected inlets from drinking water treatment plants from the Mahavali River and appropriate lab tests were performed. The results reveals a sad truth that the largest river on the Paradise has contaminations of antimicrobial resistant bacteria as well as multi-drug resistant ones too. And the interesting thing was our results are matched with the overall antibiotic usage pattern of the country. Some studies that were conducted previously says that penicillins, tetracyclines, and quinolones are the ones which are highly used antibiotics in our country in both veterinary medicine and human medicine. So, and our results are matched with these previous conducted studies. And furthermore, presence of antimicrobial resistant bacteria in river water is not a good one, because river water is not stagnant. All along its way, it can spread the antimicrobial resistance from place to place within a short period of time. So, in order to control the current situation from further worsening, further contamination should be prevented immediately. It's not a single person's job. as a part of a society, each and every one of us has some sort of responsibilities. Finally, as a famous proverb says, reap what you sow. But in our situation, I think it's most appropriate to say, we reap what we sow, because we humans are the ones who introduce the antimicrobials to the environment, and now it will be a biggest problem because, now humans are the ones who introduce the antimicrobials to the environment, and now we have to pay for it. It's a bitter truth, and I know it's harsh, but we can control it at some certain level by work as a team, as a community, as a country, as well as the world. I think it would be a correct solution for the current situation of antimicrobial resistance in all over the world. Thank you.
Thank you. And now I would like to invite Ms. S.P. Chandrasekhara, and the title of the presentation is CAV, a sneaky trigger for IBH in broilers.
Do you know that chicken is the most common protein source in the world? Yes, it is same in Sri Lanka as well. So that's why effective poultry industry is important to the country. As veterinarians, we have huge responsibility to monitor, prevent and control diseases that can cause more production losses to the field. The major threat is, one of the major threats in the poultry industry is immunosuppressive diseases such as chicken anemia virus, which mainly affect to young birds and cause anemia and thymus atrophy. Sometimes this disease can be present as subclinical disease without showing any clinical sign and cause immunosuppression and lead to other secondary diseases. Nowadays, Sri Lanka has been experiencing huge production losses due to fowl adenovirus associated inclusion body hepatitis, another viral disease in the poultry industry. So the objective of our study was to see whether chicken anemia virus is circulating in IBH positive farms in Kurunegala district, and if so, to emphasize the importance of vaccination, prevention of the chicken anemia virus in the field. So we collected samples, tissue samples from 4 broiler farms in Kurunegala district, and we choose Kurunegala because of high density of poultry farms in country, and those all samples were positive previously for IBH, and total 12 samples were subjected to PCR specific PCR for chicken anemia virus, and the interesting thing is none of those birds had shown clinical signs related to chicken anemia virus. So 75% from our samples were positive for chicken anemia virus, and at least one sample from every farm was positive for chicken anemia virus. So we concluded that chicken anemia virus can be present as a subclinical disease, present silently, affect subclinically, and undergoing unnoticeable while causing many other diseases like IBH. So it is important to maintain proper monitoring system and vaccination system for reduce further production losses. Thank you.
Thank you. And now I would like to invite Mr. De Silva, HMHG, and the title of the presentation, Divulge Fleas, a Guide to Identification.
Hello animal lovers. From the next 3 minutes, I am going to read all of your mind. To do this, I need all of you to think and remember, have you ever bitten by a flea from your dog or cat? So, you just exposed to a emerging zoonosis due to this flea bite. Yes, Sternocephalus felis like flea species are the known vector for transmitting emerging zoonosis, Rickettsia and Bartonella. So, do you know which species living in Sri Lanka? And how can we identify these flea species? That is the first question I got before these studies. So, I went through external features of fleas like comb and head shape. Then I created and developed a morphological key according to their external features. Using these, I tried to identify flea identity. So I collected 32 samples, 28 from dogs and 4 from cat, overall 190 fleas from Kandy district. Then I used my morphological key and found out. Can you guess which flea species in common? So I first say, now I'm going to read your mind. I can see some of you think it is Sternocephalidus canis because of the dogs, and some of you think it is Sternocephalidus felis. But the actual result is Sternocephalidus orientis was 72% I found on my result. Then 21% of them belong to Sternocephalidus felis, and 6% of them is mixed or hybrid morphology. So these two identified species are the known vector for transmitting Rickettsia and Bartonella. Also, they had a wide host ranges. So epidemiologically, this is a news. But we don't know about Sri Lankan situation. So when talking about the morphological keys I use, these are easy and affordable to use, but it should be updated to latest information. But for confirmation, we have to use the molecular methods. So now you can understand why I put divulge fleas in my slide. So this is starting a guide to identification of fleas. So I am going to guide you through this journey. I hope to continue these studies in future. So my take-home message is be alert and please don't get bitten by flea in future. Thank you.
Thank you. And now I would like to invite Ms. J.A.A. Sulochana. And the title of the presentation is Canine Cryptosporidiosis, Treat or Retreat.
Around 1980s, a new parasite called Cryptosporidium has been detected. A new parasite called Cryptosporidium has been detected. Since this organism has been detected in humans and many animals, including poultry, including livestock. Including livestock. Due to active searching, this organism has been detected in many countries in the world. In my study, also, we detected presence of Cryptosporidium in around Kandy district. Presence of Cryptosporidium in around Kandy district. The overall infection rate of Cryptosporidium is around 10%. And it is important to know whether, now the question is whether it is cause clinical significant disease in human or in dogs, or it is a harmless organism for dogs. So we evaluated the clinical signs of affected dogs. only 4 out of 11 positive dogs show clinical signs of cryptosporidiosis, but 3 of them were having other concurrent diseases. 1 dog had parvoviral infection and other 2 dogs had gastrointestinal parasitism. Therefore, the clinical significance of the cryptosporidiosis is still doubtful. However, all of we know the cryptosporidiosis is a clinical significant disease in humans. And it is important to know whether the Cryptosporidium species affecting dogs can cause infection in humans. Therefore, further molecular studies are required for the identify the Cryptosporidium species in dogs and humans. Otherwise, we can't justify the treatment because already we have faced a lot of problems associated with antimicrobial resistance due to unuse of antimicrobial drugs. So let's do more research on Cryptosporidium and decide whether treatment is needed or not for the dogs who have not shown clinical signs. Thank you.
Thank you. And moving to the final presentation of the day, I would like to invite Ms. SKAI Naomi, and the title of the presentation is Heifer Synchronization, Pioneering Protocols for Sri Lanka's Dairy Future.
Heifers. Heifers are the future milk productions of dairy. So, heifers should look after from the birth until they calve. Normally, heifers reproductively mature at 14 to 16 months and expected to calve at 25 to 27 months. But in Sri Lanka, that is not occurring properly. So, we decided to do two synchronization protocol for the heifers. Those are prostaglandin based breeding program and the intravaginal progesterone device and estradiol benzoate cumate program. We decide this protocol in the large scale intensively managed dairy farms in Nuwara district. We use 57 animals for this protocol, and after data collection and the analysis, the result says that in the prostaglandin breeding program, the pregnancy percentage is 54, and after the estradiol benzoate breeding program, the pregnancy percentage is 33. So, it is better to use prostaglandin based breeding program, but also it is cost effective and it is easily manipulated. But we done this protocol in large scale dairy farm. In large scale dairy farm, it is hard to detect heat and we have to do AI time to time. So, it is good to use intravaginal progesterone device and estradiol cumate program. After collections with these data, we come to conclusion and then we decide to further studies and do another synchronization programs and for the heifers. Thank you.
Thank you. We have now come to the end of the presentations. We have here with us today a distinguished guest, Dr. Roshan Priyantha, to deliver the keynote address. Dr. M.R.A. Priyantha obtained his BVSc from the Faculty of Veterinary Medicine and Animal Science, University of Peradeniya in 2001, where he then served as an assistant lecturer from 2002 to 2003. In 2008, Dr. Priyantha obtained his MSc from the Postgraduate Institute of Agriculture, University of Peradeniya. Dr. Priyantha then obtained his PhD from the Western College of Veterinary Medicine, University of Saskatchewan, Canada in 2013, and is now also a fellow member of AMR Surveillance Animal Health. Dr. Priyantha currently serves as the principal scientist of the Bacteriology and Pathology Division of the Veterinary Research Institute and also serves as the head of the Division of Bacteriology. He has also served as an adjunct professor at the Department of Veterinary Microbiology at the Western College of Veterinary Medicine, University of Saskatchewan, and I kindly invite you to deliver the keynote address.
Thank you very much. I think, first of all, thank you very much for inviting me for this event. And yes, today actually I'm going to talk about a little bit on the role of veterinarians in National Strategic Plan for AMR, because we have 2023-2028 National Strategic Plan in the country. This is a national plan, so we have to follow that. But of course, for my study, a couple of students, they came up with a nice background, so I can start from that level. Yeah, if you look at the antimicrobial resistance, it's a microbial response. So then, and also it's a natural mechanism, so we cannot avoid it. Of course, it's normally happening in the environment. And it doesn't mean that we cannot use antimicrobial in animals. It's not limited to the human. Even animals, they need antimicrobials. But of course, the pathogens, there are a number of pathogens, maybe indicator organisms, and different mechanisms, or we call it multiple mechanisms. So because of that, it has become a very complicated issue. If you look at the different antimicrobial targets, yes, of course, there are a number of antimicrobial targets here. So different mechanisms and also different resistance mechanisms. That is why people are really more concerned about this issue, different mechanisms, different overcoming mechanisms, and really complicated picture. So if I get a very simple example, this is, I think, most of the people you know, because there are a lot of distinguished microbiologists working in the faculty, Dr. Anuruddhika and Professor Anuruddhika and Professor Rasika and all this sort of, because if you look at this mechanism, so this is in your right side, you can see the beta-lactamase resistant. Because some of the veterinarians in the field, sometimes they really misidentify the difference between methicillin resistance with the beta-lactamase resistance. In beta-lactamase resistance, basically this organism excreted to the cells to the outside. This is actually extracellular enzyme. Because of this beta-lactamases, they really destroy the beta-lactam ring. Because of that, actually, they causes the resistant mechanism. But if you look at the other side, it's actually the methicillin resistant, which is caused by presence of MEK gene, or probably MEK gene, maybe sometimes MEK gene, but which causes the some of the differentiations of penicillin binding protein. So because of the changes in penicillin binding protein, penicillin cannot go inside. Because of that, all the quinolone category grafts, they cannot go inside. Because of that, that is the real mechanism. So there are two different mechanisms. These are two basic mechanisms, but there are a large number of mechanisms for that. Also, the dissemination of resistance is another issue, because even though we have different mechanisms, the bacteria, they are really smart. They disseminate between the one bacteria to the other bacteria. Their dissemination is really fast. Their dissemination very smart. Because of that, it has become an issue. So why do we worry about, as a veterinarians, about antimicrobial resistance? Because it's a veterinarian's responsibility. Because veterinarians, they have a sound understanding on the medicine and also about the microbiology and also about the treatment protocols, whatever the conditions in life science, they should have a sort of good background on life science. So that's our responsibility. And also, if you look at the veterinary sector, maybe sometimes some people talk about animal health sector. It doesn't, it consists of maybe the department, maybe regulatory body in Sri Lanka currently, but not only department, but faculty and private practitioners, maybe livestock farms, pharmaceutical industry, food industry, all the people are included, because it's not the government, it's not the private, all sort of practitioners, they are really included. The important thing is that we all bound to the health regulation, that is the international health regulation, or maybe sometimes we call it joint external evaluation, all the human health counterparts and veterinarians, we all bound. So we have to do sort of necessary action, the combating antimicrobial resistance. It's by law, so we cannot avoid that. That is why we really more concerned about, and one of the reasons to concern about the antimicrobial resistance. Not only from the international health regulation, there's another mechanism we call performance of veterinary service evaluation, which is actually coordinated by the WUHA, World Organization of Animal Health. It is also there. So then because of all these regulation, international counterparts we have to do. That's why we have to fight against antimicrobial resistance. This is the last time, this year actually, last month, our antimicrobial resistance from the health sector and also from veterinary sector, agriculture, all the sectors were really evaluated by the Dr. Sebastian Heller from the Robert Koch Institute, Germany. He's the one who evaluated and came up with the next 5 years plan for the antimicrobial resistance in the country. So if you little bit about, because we had actually Sri Lanka is one of the country, we had a very good national strategy plan for combating antimicrobial resistance, which was 2017 to 2020. But unfortunately, because of the pandemic, COVID-19, and also the followed by economic crisis, I think we have very limited success, or even in human health sector, and also the animal health sector, very limited success, because we don't have a very good success behind that. This is the National Strategic Plan for the Combating Antimicrobial Resistance, which was issued by the Ministry of Health. Then now actually the Ministry of Health and all the veterinarians and Department of Agriculture and Department of Fisheries and Ministry of Health, Ministry of Environment, they all came up with now they are working on the new proposed, new national strategic plan. We call this proposed national strategic plan for 2023-2028. So we usually this consists of 7 priority areas. Usually the previous one actually we didn't have environment safety, because environment section was not included in the previous action plan, but it has been addressed. But the important thing is that all these sectors, veterinarians, they have a role. So that is why I'm going to highlight here. If you look at the first priority area, that is organization of One Health AMR response, still we don't have it. Yes, we have been discussing quite long years, but still mechanism not there. But of course we have to go for that. For example, the human health, yes, they do a lot of antimicrobials, and animal health, fisheries, they use that, agriculture, usually they do a lot of antifungal, and environment, basically hospital waste, clinical waste, they're really more concerned about that. So of course we cannot work alone now. We have to get together, and we should have mechanisms to share our experience within this existing system. That is why this national strategic plan, they are going to highlight on One Health AMR response rather than going for the individual response. And all the other important factors, actually this strategic plan, they are going to highlight the awareness and understanding of AMR. But of course, I think the comprehensive and sustained One Health national awareness program on AMR, which is the requirement currently. Of course, the veterinarians, they have a very good understanding about the antimicrobial resistance, but the problem in Sri Lanka, in our sector, the veterinary paraprofessionals, because they don't have much understanding on that. Because sometimes veterinarians, they don't want to do that, so that's actually lagging behind. That is one of the reasons that we have to overcome this area. Not only the understanding the curriculum, but of course the CPD program, continuous professional development programs, are required for the veterinary paraprofessional as well. And these are some of the awareness programs, some of the Professor Rubini Kalupan and all the people. And the other important factor in strategic plan, they really highlight the awareness. Because of course the faculty, they maybe have your individual study, but maybe the VRA, we have another individual study, but it doesn't work for us, because we should have a national resistance surveillance system. The human health sector, they have that, including 26 sentinel sites, but still the veterinary sector, we don't have it. So that's what the documents really highlight the importance of this kind of a surveillance system. So maybe all the veterinarians, maybe in the faculty, maybe in any other institutes, or maybe government, we have to come up with the national surveillance. Maybe we're looking at the pathogenic organism or the indicator organism, but still we should have a national resistance surveillance. And the other important factor is the antimicrobial resistance, antimicrobial residues. Of course, the faculty, Dr. Ruchika's lab, they actually, even this time, we really appreciate your all, because in country roles, you are the only one laboratory in Sri Lanka with the accredited facility for the detecting antimicrobial residues. Yeah, thank you for Professor Ruchika for all the hard work. So it's really appreciate, but we need a surveillance system with antimicrobial residues, because since we don't have a currently system of surveillance, yeah, we do have to get a sampling, but still surveillance system is lagging behind. So that's what really highlighting. And also the antimicrobial consumption, because we import antimicrobial from some other countries. So because of that, usually we all get all the information on import data. Usually we submit to the OIE annually or sort of antimicrobial. For example, in this example, so 2016, since macrolide is maybe around 1 ton, maybe 10 tons, but you see the 2019, so we know 42 tons of macrolides. That's maybe because of the Mycoplasma synovial infection. All these information are really recorded. So these data actually really valid reasons. Then only we can decide where are we. I mean, we can get a policy decision based on that. So that's why the burden of assessments of AMR is really important. And these are the information from every year, all the important, all the categories, because actually in the Veterinary Drugs Control Authority, we monitor all the antimicrobials in veterinary sector, like NMRA in human sector. And also the other area that actually we are going to highlight, because we have a chain of veterinarians and a lot of private practitioners and maybe private hospital, but still we don't have the IPC policy for companion animal. So which has been really highlighted in the document, because we need to have a IPC policy, infections prevention control policy for companion animal and livestock production system, and also aquaculture. Because I think the veterinarian role is to work on aquaculture as well, based on this one. And also the IPC program, because we don't have IPC program currently. And not only the IPC policy, but also the IPC program, but we also need monitoring mechanism to IPC practices in animal, companion animal, livestock, and also aquaculture. Those are the other priority areas we have to work on that. And also the best management practices for animal health and aquaculture, still we are lagging behind. But of course, the last year, the SLS had started this process. Maybe next year we will get these GMP practices, ISO standard for the all sort of livestock species. But of course, identify the research priorities. Maybe the university animal health sections. I mean, you do have your all research program, but as a country, we don't have a country priority. Maybe CARPE, they have a priority, but but this document really highlights the research priorities on infectious diseases, which is one of the areas that we have to work on within next 5 years as within this plan. And also the national implementations of national waste management policy, because still we have a sort of different clinical practice, even government hospital, but still we don't have a national waste management policy on veterinary sector. So it is also common. So maybe hospitals, labs, farms, aquaculture, aviaries and feed mill, yeah, whatever the things, but we should have a national policy. And also this guideline, this national strategic plan, they're really concerned about the guideline for building construction in hospital setting in relation to IPC in veterinary hospitals. But of course, the human hospital, they do have this kind of practice, but unfortunately, we don't have that. But we have to overcome within this 5 years. And also the other area, actually the priority, fifth priority area, optimizing the antimicrobial use. Especially regulate, if you look at the antimicrobials in Sri Lanka, we have a regulatory authority. We regulate and monitor registration, waiver of registration, manufacturing, importation, labeling, advertising, but all sort of things are actually we have a regulation. But unfortunately, we don't have regulation for distribution and transport, storage, sales, and disposal of antimicrobial products. So this is the area. So we have to, maybe next 5 years, we have to really work on that. And also the quality of antimicrobials, because especially people, maybe clinicians, maybe practitioners, they come up with this antimicrobial is not really in good condition. So in that case, actually, we don't have a established laboratory system currently to have a proper quality assurance of existing antimicrobial resistance, also really highlighted. And the other important thing is actually post-marketing surveillance of antimicrobial and also antimicrobial stewardship program, because in companion animal practice has been really developed, especially the most of the private sector contributions really immensely improved, but still we don't have a national antimicrobial stewardship program. So it's also important area that we have to look at carefully. Probably all the clinical microbiologists and experts from the faculty, hopefully they will support us to develop this policy within next 5 years. And then and also the monitoring of usage of antimicrobials, because antimicrobial consumption and antimicrobial usage is quite different stimuli in veterinary medicines. There are some differences, but in these 5 years actually we are more concerned about antimicrobial residues, but not animal usage, because in practical it's quite challenging for us to go to the field level to work on antimicrobial usage within this 5 period. But of course we have, as Department of Animal Production, and of course we have accepted the codex limits as the maximum residue limits for the veterinary drugs, but then we have to have implementation program, and also we have to come up with the regulatory programs based on risk-based assessment. And also the research and innovation is another area, because of sometimes we will not have a opportunity to have new medicine, but of course we can go for the different diagnostic tools and also different vaccines. Actually we produce 12 different number of vaccines, so but we have to come up with the new vaccines, maybe control the bacterial infection, maybe viral infection, because then we can minimize the usage of antimicrobials in other secondary infection. So those are the areas actually we have to think, because in the as veterinarians, this is our responsibility to come up with for the next 5 years. And if I look at my short summary, so antimicrobial resistance is a burning issue. So all the students, they came up with the nice presentation, so we have to accept that. And as veterinarians, of course, we have a major role, since we can understand, we can feel the situation. But we cannot do alone. We have to get together with all the sectors there. And this is a very good time to come up with the IPC programs and antimicrobial stewardship programs, maybe national resistance surveillance, basically integrated surveillance, and antibiotic residues monitoring mechanism, also the animal waste or good animal husbandry practices, and maybe as regulations of quality and antimicrobial distribution. So all these sectors like maybe livestock, companion, aquaculture. So next 5 years, 2020 to 2028, we have to complete all sort of things. Yes, we do discuss about antimicrobial. We have to come up with all these accepted content in this national strategic plan, because it is a national plan now. We have to contribute it. So that is the only way that we have to fight against this emerging issue. I think this is done, so thank you very much. I hope you get something. Thank you very much. Yes.
Thank you, sir. I now kindly invite the Dean of the Faculty of Veterinary Medicine and Animal Science to present a token of appreciation to Dr. M.R.A. Priyantha. Thank you, sir. And as we are expecting the results, we would kindly appreciate your patience with us. I would now take this opportunity to invite Dr. Lalante De Silva, the chairperson of the VMEU, to announce the winners of the 3-minute presentation round held today. And I also cordially invite the Dean of the Faculty of Veterinary Medicine and Animal Science, Professor P.G.A. Pushpakumara, to present the certificates to the winners.
It's my honor to announce the award winners of this undergraduate research symposium. Best oral presentation, 3rd place, Ms. S.P. Chandrasekhara. Best oral presentation, 2nd place, Mr. U.D. Jayasekara. Best oral presentation, 1st place goes to Mr. H.M.H.J. De Silva. Now I would like to announce the Merit Award winners. Mr. W.A.S.D. Veerasurya. Ms. W.U. Kumudumali. Mr. G.T.V. Ananda. Ms. D.A.H.Y. Senarath. Mr. J.J. Wesley. Mr. K.A.L. Pium. Ms. G.I.U.S. Satsarani. Mr. S. Tanish. Ms. J.W.Y. Senaviratna. Mr. J.W.Y. Senaviratna. Ms. S. Tirutanigasalam. Ms. J.A.A. Sulochana. Naomi. Congratulations for all of you. Thank you.
Thank you, sir and madam. We have now come to the end of the proceedings. I would like to invite Dr. Lalante De Silva to deliver the vote of thanks.
Thank you, Kajini. Vice Chancellor, Deans of the Faculties, Dean of the Faculty of Veterinary Medicine and Animal Science, Heads of the Departments, Academic Staff Members, Students, distinguished guests, and all the other participants. Ladies and gentlemen, it's a great honor to deliver the vote of thanks today. First of all, I would like to express our heartfelt gratitude to the chief guest of our event, Vice Chancellor, Professor M.D. Lamawansa, and also the Deans of the Faculties and all the other distinguished guests for your presence here. Your presence here made this event an honorable occasion. I would also like to express our deep appreciation to Professor and guidance in organizing this event in a successful manner. Moreover, I would like to extend my heartfelt thanks to our distinguished keynote speaker, Dr. Roshan Priyantha. The shared experience, expertise, and insights enriched this event a lot. And also, our esteemed panel of judges deserves special recognition for their invaluable contribution in selecting the best presenters. Dear Sir, Madam, thank you very much. I would like to acknowledge the Senate Research Council for their generous financial support, and also I express our gratitude to Professor Ruwani Kalupahana, Chairperson of the Faculty Research Committee, for her efforts in coordinating funding for this event. A special mention goes to Dr. Sampath Lokugalapatti, the Chief Editor, and also the entire editorial board of the proceedings of the 3rd Undergraduate Research Symposium. Because of your efforts, we were able to prepare this abstract book of 80 undergraduate research abstracts. What we're witnessing today is the outcome or the success of tremendous efforts made by the undergraduate students, their supervisors, as well as reviewers. So let me thank all the students of these undergraduate research projects. I wish to acknowledge and thank the entire Veterinary Medical Education Unit Committee for their leadership in organizing this momentous event. The smooth conduct of 80 research presentations across 6 different parallel sessions, and also today's 3-minute presentation would not have been possible without the tremendous support extended by the Dean of the Faculty, heads of the departments, all the academic staff members, non-academic staff members, as well as the team of the English Language Teaching Unit, Assistant Registrar, Dean's Office staff, and all the faculty staff members. So thank you very much. We are also grateful to the University Webmaster, Media Unit, and the Audiovisual Technical Officers of our faculty, as well as other faculties who are joining us with today to make this a successful event. A special thank goes to University Landscape Division for their creative decorations of today's event. And I would like to express our appreciation for our talented compeer, Ms. Kajini. Last but not least, I would like to express for this 3rd Undergraduate Research Symposium today. Let me conclude my vote of thanks with this quote: "Research is the compass that guides us through unrevealed territories, and the teamwork is the wind in our sails that propels us forward towards discovery." Thank you very much.
Thank you. Now I would like to invite Professor P.G.A. Pushpakumara to launch the proceedings of the 3rd Undergraduate Research Symposium. Thank you, sir, and please rise for the national anthem.
Sri Lanka Mata, Appa Sri Lanka, Namo 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 Mata. Sundara Sivarini, Surani Ati Sobamana Lanka. Ranya Kanaya Mekamal Balatulu Iri Jaya Bhumi Ranya. Appa Hatta Sapa Siri Setta Sadhana, Jeevanaye Mata. Viliganu Mena Appa Bhakti Puja, Namo Namo Mata. Appa Sri Lanka, Namo 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 Mata. Obame Appa Vidya, Obamaya Appa Satya, Obame Appa Shanti, Appa Hadatula Bhakti, Oba Appa Aloke, Appa De Anuprani, Oba Appa Jeevanane, Appa Muktiya Obade. Nava Jeevana Devini, Nithina Appa Pubudu Karan Mata. Jnana Pida Vadavanina Ragena Yanu Mena Jaya Bhumi Kara. Ekkamavaka Kedaru Kala Vadina, Yamu Yamu Vinopama. Prema Vadasana Veda Durana, Namo Namo Mata. Appa Sri Lanka, Namo 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 Mata.
Thank you for joining with us today. Hope you all had a fruitful afternoon. This is to kindly remind you all that the refreshments will be served at the dining hall of the Dean's office. Thank you.